Hello students, today I am going to start a new topic called Metabolism of Xenobiotics. Greek word xenos means stranger. A xenobiotic is a compound that is foreign to the body. The principal classes of xenobiotics of medical relevance are drugs, food additives, pollutants, chemical carcinogens, and various compounds that have found their way into a environment by one route or another, such as polychlorinated biphenyls and certain insecticides. More than 2 lakhs of manufactured environmental chemicals exist. And how these how does the body handle foreign compounds? Even though all tissues metabolize xenobiotics, the liver is the major organ involved. About 30 different enzymes are involved in the metabolism of xenobiotics. Usually, usually the metabolism of xenobiotics is considered in two phases. Phase 1 reactions. The major reactions involved is hydroxylation or oxidation, catalyzed mainly by the members of a class of enzymes referred to as monooxygenases or cytochrome P450s. Hydroxylation may terminate the action of a drug, though this is not always the case. In addition to hydroxylation, these enzymes catalyze a wide range of reactions, including those involving deamination, dehalogenation, desulfuration, epoxidation, peroxidation, and reduction. Reactions involving hydrolysis, such as that's catalyzed by esterases, and certain other non-P450 catalyzed reactions also occur in phase 1 reaction. And another is the phase 2 reaction. In phase 2 reactions, the hydroxylated or other compounds produced in phase 1 are converted by specific enzymes to various polar metabolites by conjugation with glucuronic acid, glutathione, sulfate, glycine, and also by other mechanisms like acetylation and methylation. The overall purpose of the two phases of the metabolism of xenobiotics is to increase their water solubility, that is polarity, and thus excretion from the body. Okay. Now, very hydrophobic xenobiotics would persist in adipose tissue almost indefinitely if they were not converted to more polar forms. In certain cases, phase 1 metabolic reactions convert xenobiotics from inactive biomolecules to the biologically active compounds. The term detoxification is sometimes used for many of the reactions involved in the metabolism of xenobiotics. The term detoxification is not exactly synonymous to the metabolism of xenobiotics because not all the reactions uh, not all reactions are not detoxifying. This is why the term biotransformation is more appropriate. In a few cases, the reactions involving the metabolism of xenobiotics could turn them more active and sometimes even toxic. This is true in case of procarcinogens, which are converted to potential carcinogens after being metabolized. Now come to the phase 1 reactions. Phase 1 reactions, the most important uh, reaction is the hydroxylation or oxidation. Xenobiotics are metabolized mostly by hydroxylation or oxidation during phase 1 reaction. The reaction catalyzed by monoxygenases, that is cytochrome P50, as follows. You see the XH, that is the xenobiotic in these reactions, and you see this oxygen and the NADPH with the help of in the presence of NADPH and hydrogen ion. That reaction is catalyzed by this cytochrome P450. Okay, so one of the oxygen atom that is introduced, okay, that is utilized, okay, and these enzymes are called the cytochrome P450, that's why called monooxygenases. Okay, now this a very wide variety of xenobiotics, including drugs, carcinogens, pesticides, petroleum products, and pollutants, such as mixture of this poly chlorinated biphenyls. They are, biotransformed. In addition, endogenous compounds such as certain steroids, eicosanoids, fatty acids, and retinoids are also the substrates, okay, which are substrates for the cytochrome P450s. Okay. The substrates are generally lipophilic and they are rendered more hydrophilic by hydroxylation reaction. The responsible enzymes are called monooxygenases or cytochrome P50. Cytochrome P450 is considered the most versatile biocatalyst known. It has been shown by the use of that is we have mentioned already that one atom of oxygen enters this xenobiotic and one atom enters the water. This that's why the dual fate of the oxygen accounts for the this form naming of these monoxygenase as mixed function oxidases. And the following are the important point concerning the cytochrome P450s. Okay, cytochrome P450s, we have mentioned that human genome expresses a large number of isoforms, about 150 of cytochrome P450. The enzymes which are structurally and catalytically identical but have distinct genetic origin are called isozymes or isoforms. The major cytochrome P450s in the drug metabolisms are members of cytochrome P1, cytochrome P2, and cytochrome P3's families. Okay, like hemoglobin, these cytochromes are they're hemoproteins. Okay, so they are hemoproteins, and they are widely distributed across species, including their bacteria, and they are present in highest amount in the liver cells and enterocytes, but they are probably present in all tissues. In the liver and most other tissues, they are present mainly in the membranes of the smooth endoplasmic reticulum, which constitute the part of the microsomal fraction when tissue is subjected to subcellular fractionation. In hepatic microsomes, cytochrome P450s can comprise as much as 20% of the total protein. P450s are found in most tissues, though often in low compared with the liver. In the adrenal, they are also found in the mitochondria as well as in the endoplasmic reticulum. The various hydroxylases present in that organ play an important role in cholesterol and steroid biosynthesis. The mitochondrial cytochrome P450 system differs from the microsomal system in that it uses a an NADPH-linked flavoprotein called adrenodoxin reductase, and a non-heme iron sulfur protein called adrenodoxin. In addition, the specific P450 isoforms involved in steroid biosynthesis are generally much more restricted in their substrate specificity. At least 6 different species of cytochrome P450s are present in the endoplasmic reticulum of human liver, each with a wide and somewhat overlapping, each with some, you know, wide and somewhat overlapping substrate specificities, and acting on both the xenobiotics and endogenous compound. Okay. NADPH, we see this, and in genes for many isoforms. The genes for many isoforms of P450 from both humans and animals, such as rat, has been isolated and studied in detail in many years. The combination of there being a number of different types of these cytochrome P450 and each having a relatively wide substrate specificity explains why the cytochrome P450 family can metabolize the thousands of different chemicals. And here, this in the reaction, we have seen that NADPH, not NADH, and that is involved in the reaction mechanism of the cytochrome P450. Lipids are also the components of the cytochrome P450 system. The preferred lipid is the phosphatidylcholine, which is the major lipid formed in the membranes of endoplasmic reticulum. And most isoforms of cytochrome P450 are inducible. For example, administration of the phenobarbital or many other drugs causes hypertrophy of the smooth endoplasmic reticulum and 3 to 4-fold increase in the amount of cytochrome P450 within 4 to 5 days. The mechanism of induction has been studied extensively and in most cases involves the increased transcription of mRNA for cytochrome P450. However, certain cases of induction involve stabilization of the mRNA, enzyme stabilization, or other mechanisms such as an effect of translation. Induction of cytochrome P450 has important clinical implications since it is a biochemical mechanism for drug interaction. A drug interaction has occurred when the effects of one drug are altered by prior, concurrent, or later administration of another drug. Now, the situation, suppose in a patient taking an anticoagulant like warfarin to prevent the blood clotting, and this drug is metabolized by cytochrome, suppose P2 and C9, a type of isoform of cytochrome. And concomitantly, the patient is started on phenobarbital, that is a drug that is used to treat certain type of epilepsy, and that phenobarbital is an inducer of cytochrome P450. But at the same, so as the drugs warfarin and phenobarbital at the same time they're used, and phenobarbital is an inducer. If the dose of warfarin is not changed, what happens? After 5 days or so, the level of that cytochrome P2C9, that is isoform, in the patient's liver will be elevated 3 to 4-fold. And this in turn means that warfarin will be metabolized much more quicker than before, and the dosage will have become inadequate. Therefore, the dose must be increased if warfarin is to be therapeutically effective. And for this purpose, and the problem could arise also in later if the phenobarbital is discontinued, the reverse thing happen, but the increased dosage of warfarin stays the same. the patient will be at risk of bleeding since the high dose of warfarin will even more active than before. Okay, so that is a type of drug interaction can occur. Okay, now the second type of reaction, okay, what you see the reduction, another type of reaction in phase 1 reaction is a reduction. The nitro compounds is a, are reduced to their uh, amines and aldehydes. Aldehydes and ketones are reduced to alcohol, such as nitrobenzene to aminobenzene, picric acid to picramic acid, okay, paranitrophenol to para-aminophenol, and the reaction is the hydrolysis. Hydrolysis, many of the toxic molecules in the body are broken down into smaller molecules by hydrolysis. You see this XY, that is the xenobiotic here with reacts with water to form XOH and Y. The hydroxyl group and the hydrogen ion of the water are transferred to these different fragments of the substrate XY. Bonds such as glycoside, ester, amides, and so on are biotransformed by hydrolysis with hydrolysis, and aspirin, the popular analgesic, is metabolized by hydrolysis also. Other examples include atropine, xylocaine, acetanilide, and procaine. Now come to the phase 2 reactions. A xenobiotic metabolized into a polar compound in phase 1 reaction may not be sufficiently polar to be eliminated easily from the body. Usually, these intermediates need to undergo more biotransformation reactions. Phase 2 reactions, mostly conjugating reactions, metabolize these hydroxylated byproducts into even more hydrophilic molecules, and they are eventually excreted in the urine or bile. The conjugation involves the addition of body metabolites to xenobiotics, rendering them more polar, non-toxic, and easily excretable compound. Most of the conjugating agents act on xenobiotics which have already undergone biotransformation by phase 1 reactions. However, certain xenobiotics, such as phenol, are directly conjugated without undergo. So, certain xenobiotics, such as phenol, are directly conjugated without undergoing phase 1 reactions. Now, the following are the endogenous substances act as conjugating agents. One is the first the glucuronic acid. Glucuronidation catalyzed by the glucuronyl transferase, okay, is the most frequent conjugation reaction. Okay, conjugation reaction, and UTP glucuronic acid is the glucuronyl donor. We have seen this reaction, the UDP glucuronic acid and the bilirubin here is the which is conjugated and form the bilirubin diglucuronide and the conjugated bilirubin, and that becomes more water soluble. UDP gluconyl transferase is the enzyme this catalyzes this reaction. So it is non-polar bilirubin is converted to this polar bilirubin diglucuronide that is more water soluble and that is excreted in the bile. Like aniline, meprobamate, a tranquilizer, morphine, the analgesic, several steroids, acetyl aminofluorine, a carcinogen, all are excreted as their glucuronide derivatives. Okay, sure. And second type of reaction, what is called is the sulfation, mm, sulfation reaction. Donor here, some alcohols you know, aryl amines and phenols are sulfated, and the sulfate donor is in the and other biologic sulfation reactions like sulfation of steroid, glycosaminoglycans, glycolipids, and glycoprotein is the 3' phosphoadenosine 5' phosphosulfate, also called PAPS, and this compound is called the active sulfate, 3' phosphoadenosine 5' phosphosulfate. See, this phenol is, that undergoes the sulfation to form this phenyl sulfate and PAP. Next is the glutathione. So glutathione is the is a glutathione. It is a tripeptide. It is called gamma. That is gamma glutamyl cysteine glycine. That means glutamate, cysteine, and glycine. And the three amino acids form this tripeptide. And the conjugates a number of xenobiotics, including certain carcinogens. The enzyme catalyzing this reaction is the glutathione S transferase and occur in high concentration in the liver. Glutathione is commonly abbreviated as GSH, and that because the sulfidyl group comes from the cysteine, that is the amino acid that is present in the glutathione, and which actually takes part in this reaction. So cysteine component of glutathione usually detoxifies the alkyl or aryl halides, alkanes, and epoxides by conjugation. Conjugation of the anti-cancer drug cyclophosphamide by glutathione is also a well-known example. And fourth is the glycine. Okay, as a part of the normal body metabolism, glycine conjugates the bile acids, like cholic acid, chenodeoxycholic acid. It also uh, detoxifies many other substances such as benzoic acid, that is a food preservative. Benzoic acid is converted to hippuric acid. Cholic acid is converted to these bile acids. Cholic acid, chenodeoxycholic acid. They are converted to glycocholic and glycocenodeoxycholic acid. And the benzoic acid, the food preservative, that is converted to hippuric acid. Next is the acetylation. Acetylation is represented by, okay, you see this xenobiotic X is the xenobiotic, you see, and enzyme is acetyl transferase, and acetyl CoA is acetyl donor in these acetylation reactions, and these reactions are catalyzed by acetyl transferase. A number of drugs are detoxified by acetylation. The drug isoniazid used in the treatment of tuberculosis is subject to acetylation. Methylation. Methylation, methyl transferase enzymes catalyze the reaction, uh, and you see this S-adenosylmethionine, it is also called active methionine, acts as the methyl donor. So here in this reaction, it's a xenobiotic XOH, and that S-adenosylmethionine, the active methyl, that is the methyl donor, active methionine, the donors methyl group, and that forms the S-adenosyl homocysteine, and xenobiotics is detoxified. Okay. Now, the activities. Now, the activities of the xenobiotics, metabolizing enzymes are affected by the age, sex, and other factors. Various and factors that affect the activities of the enzymes metabolizing xenobiotics. The activities of these enzymes may differ substantially among the species. There are significant differences in enzyme activities among the individuals, and many of which appear to be due to genetic factors. The activities of some of these enzymes vary according to the age and sex. Intake of various xenobiotics such as phenobarbital, polychlorinated biphenyls, or certain hydrocarbons can cause enzyme induction. It is thus important to know whether or not an individual has been exposed to these inducing agents in evaluating biochemical response to the xenobiotics. It is always important when taking a clinical history to ask whether the patient has been taking any drugs or other therapeutic preparations. Metabolites of certain xenobiotics can inhibit or stimulate the activities of xenobiotic metabolizing enzymes. Again, this can affect the doses of certain drugs that are administered to patients. Various diseases such as cirrhosis of liver. So because liver is the main organ for metabolism of xenobiotics. So the diseases of the liver like cirrhosis of liver can affect the activities of drug metabolizing enzymes, sometimes necessitating adjustment of dosage of various drugs for patients with these disorders. So this completes the topic, and thank you.